Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Please type any scientific questions in the Zoom Q&A box. Please do not type your questions in the chat box. I'll ask you to redirect those to the Q&A box. All attendees can prioritize questions by clicking on the thumbs up/like button. Questions will be responded to at the end of the presentation. Closed captioning is available. The link will be placed in the Zoom chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to Dr. Anthony Dickherber, Program Director in the National Cancer Institute's Office of the Director, Center for Strategic Scientific Initiatives, and Director of the NCI Innovative Molecular Analysis Technologies Program. Tony?
Thank you, Alan. And I think I can go ahead and share my slides. Okay, I hope that's coming through. So welcome everyone to NCI's Cancer Moonshot Seminar Series, which is dedicated to highlighting progress on the array of projects that were initiated in response to the 10 recommendations that guide NCI's Cancer Moonshot Initiative. Today's lecture is aligned with recommendation J for new enabling technologies, and I'm here to provide some context and background before handing over to our moderator to introduce our speaker. So I'll start with a quick reminder that these recommendations were developed by a blue ribbon panel comprised of 28 leading cancer scientists, clinicians, and patient advocates, which was subdivided, as you see on the slide, into seven distinct working groups made up of well over 100 thought leaders in relevant fields that ultimately formulated the 10 recommendations that guide the Moonshot Initiative. By and large, each of these working groups provided described priorities that developed into 9 of the 10 challenges, 10 recommendations. But recommendation J, by contrast, was a recognition that in the discussions across all of the working groups, there was a recognition of the need to leverage the state-of-the-art technologies to the maximum extent possible and do what we can to accelerate the availability of emerging technology platforms that could contribute to accelerating progress across the diversity of recommendations. And I want to make sure everyone has a sense for the diversity of recommendations we got. What I'm highlighting here is just snippets, excerpts from the different working group reports that called out specific technology opportunities that really needed to be advanced. And suffice it to say, we had a lot of different technology fronts upon which to try to focus. But what we did was, in one of the very first responses to the Cancer Moonshot, one of the first funding opportunities was one in which we called all of the categories or all of the different recommendations for technology advancement into about 4 different bins and requested for projects from the community. And so the talk you'll hear today was one of the few awards that we were able to make to that very impressive response from the community. And in this case, for advancing technologies that will overcome some of the complexity of cancer in the context of clinical diagnostics. So I'd like to go ahead and hand over to our moderator, Dr. Richard Levinson, who is an avid technology developer himself, especially given that he's a pathologist that likes to spend some of his spare time teaching pigeons to read slides better than his colleagues do. But with that, I leave it with Dr. Levinson to take over from here.
Thank you so much, Tony. It is indeed my pleasure to introduce Professor Herr, who's from Berkeley, my hometown. And what I'll do is very briefly touch on some of her background and then leave the rest of her introduction to herself, since I think she knows herself better than I do. So she is a professor of bioengineering at Berkeley, and she got her BS degree at California Institute of Technology, and then mechanical engineering at Stanford University. She's also a Chan Zuckerberg BioHub investigator, and her research interests, I'm sure, will be touched upon in her talk, but they include bioinstrumentation innovation to advance quantitation in the biosciences and electrokinetic phenomena in single cell and subcellular analyses. And so I think you've heard enough from me. I'm very happy to hand it over to Professor Herr.
Thank you so much. Thank you very, very much for the invitation to be here today. And for those of you who are spending your time with us, either in person at your location or videotaped afterwards. So thank you so much to everyone for the invitation to join and for this opportunity to work in this absolutely essential area to moving new enabling technologies forward. Today, I step back a little bit and wanted to give you a talk that centers more on what we're seeing as advances in mapping cellular to molecular heterogeneity. And one of the technologies that I'll introduce today is this 3D projection electrophoresis format that we've been pushing forward. So before I begin, I did want to take a moment to thank my research group, their photo here. We are in the Department of Bioengineering at UC Berkeley. Also, I'm a faculty scientist at Lawrence Berkeley National Labs, and as was mentioned, an investigator in the Chan Zuckerberg BioHub. If you have any follow-up questions that we don't get to today, please feel free to send me an email. My email address is at the bottom of this slide and also on our department webpage. I am always very interested to hear constructive criticism as well as new opportunities for helping to surmount measurement gaps, especially in this cellular to molecular area that I'll talk about today. So I hope to hear from some of you for sure. So zooming down a little bit in terms of the topic that I'll describe today, I did want to call out a couple of individuals, thank them very much for the work that they've done, our collaboration together, and providing me the opportunity to share some of that with you today. I also would be remiss if I didn't explicitly thank the NIH Cancer Moonshot program. Thank you for the observations and the assessments that were done, as well as the opportunity to bring in researchers to help start moving towards solutions in these vast and diverse areas that were identified. I also want to thank NIH for supporting some of the fundamental research that builds the foundation for what I'll talk about today. And as an engineer, a bona fide engineer by training, I have to say a special thank you to the National Institutes of Health for the foresight to understand that measurement technology makes understanding possible. And so we see our work as very much enabling as the ultimate goal for the work that's being done both in the basic sciences and clinical medicine as well. So thank you so much for that foresight. We know with recent advances in our approach to biosciences and biomedicine that understanding cell-to-cell variation is important to many questions involving cancer. We also know that there is an importance in not just cell-to-cell heterogeneity and defining the characteristic we're interested in understanding and then assessing that characteristic, the variation of that characteristic. But we also know that molecular variation is important. I would argue that there have been tremendous advances in our understanding of biosciences and biomedicine and cancer in particular over the past couple of decades because of a focus on single cell resolution. Most of that understanding has come from an explosion of new technologies and measurement systems that are focused on single cell resolution at the genomic and transcriptomic level. And so this is important because we know that the molecular machines of the cell, if you will, the active effectors of function, proteins in individual cells, some of the information for the repertoire of proteins that are being expressed and functional at any one time is of course encoded in the genome and at the transcriptome level. But we also know that there is a constellation of information that cannot be tapped with single cell measurements of the genome and transcriptome. So we are literally blind to what is being referred to recently, and I find a very helpful term, as proteoforms that are again affecting the function and making the cellular level decision making. So we oftentimes think of protein isoforms as important indicators of protein function or basis for protein function. We also know that there are numerous site-specific features, including common post-translational and important post-translational modifications like phosphorylation and glycosylation. There are hundreds others as well. But when we put our understanding of endogenous proteins in this context of proteoforms, we can really start to appreciate the vast physical chemical diversity of those molecules, those protein molecules, as well as the combinatorial, as I mentioned, explosion of the different individual protein molecules that could be expected to be expressed in an individual cellular system at any point in time. So when we refer to proteoforms, we are really seeking to capture this combination of different forms of proteins that can be expressed, as well as these different modifications that can happen, again, in a combinatorial way across these molecules. So I like to use the phrase at the end as an inspiration, as well as a little bit of a daunting challenge. It's much more poetic than words I'm finding this morning. But that is from one of the works that really stepped forward and posited that this idea of let's capture the diversity and the dynamic nature of the proteins that we expect to be important to disease by using this word proteoform. And they see this concept as beginning to encompass this complex and, key to me, largely uncharted world of natural proteins. And so that is what my research group looks at, and that's what we've worked under the auspices of this grant to develop measurement technologies for as well. Of course, these proteoforms, these different types of protein forms that we can expect to be expressed, have a direct link to important roles in cancer. Numerous ways. I mentioned site-specific modifications, and certainly modifications like phosphorylation are important to cross-signaling more broadly. But I do want to draw your attention to a class that we'll focus on today, a class of proteoforms relevant to cancer. And that is really this idea that different types of proteins that are important, like HER2, ERBB2, estrogen receptor, and others, can manifest as something that we call a truncated isoform. So the specific molecular example I'll give you is HER2, ERBB2. It is often manifest as a large 185 kilodalton protein that spans the cellular membrane, comprised of an extracellular domain and an intracellular tyrosine kinase domain. That's important both to the function of the molecule and also the response to targeted cancer therapy. So an example that I give here is trastuzumab or Herceptin, humanized monoclonal antibody that targets the extracellular domain of this 185 kilodalton HER2 protein. As we'll get into a little bit later, HER2 has different truncated isoforms that are exhibited. We call those T-ERBB2 or truncated isoforms. The word truncated is used as an adjective here because those proteins are unfortunately missing the extracellular domain, that domain that is targeted by trastuzumab. So it's important for us to understand not just that the protein HER2 is expressed in a particular cell, but we're very interested in understanding what form of HER2 is expressed, because obviously the different forms and the stoichiometry of expression affects things such as the response to therapy, but also our understanding of how cancer cells, HER2 positive cancer cells, can start to develop resistance to therapy as well. And I'll keep it short for today because I want to focus on the technology, but this question of the stoichiometry of the proteoforms being expressed in a given cell and constellation of cells is important beyond HER2 as well. So just a little bit of an context there for the work that we're doing and why it's relevant to this program. So these truncated isoforms and proteoforms more generally exemplify what I see as unmapped areas as we talk about things like mapping or creating atlases of biology and disease. And of course, a big interest in atlas creation is at the single cell level. So as an engineer, first of all, I want to understand a little bit about what tools exist right now. Again, if we can't measure it, we can't understand it. And then also where is the gap that this project is working to fill. So sequencing, I mentioned a massive explosion in terms of single cell resolution sequencing that's available, super important in terms of our understanding of the natural world around us. Just to remind you, information that is encoded in DNA and then RNA does not reflect the dynamic state of the protein expression. Modifications or complexing, this idea of proteoforms that I mentioned a moment ago. Mass spectrometry, an impressive suite of powerful technologies that is really pushing our understanding forward. At the single cell resolution level, there have been impressive developments over the recent past, very recent past, as a matter of fact. Most of those developments with single cell measurements have been in bottom up mass spec. When we're thinking about measuring intact proteins, which is absolutely crucial to be able to, again, maintain the stoichiometry of these different proteoforms that are being expressed. We have headway to make there in terms of intact protein measurements. I'm confident that there will be and continue to be amazing advances in this instrumentation class that move us forward. But right now, detection limits, throughput for single cell measurements, and also this important characteristic of maintaining stoichiometry for those proteoforms is lost. Lastly, I would be quite remiss if I didn't pay homage to a much cursed and also much used measurement technology, and this is the immunoassay. Certainly everything from flow and mass spectrometry all the way to ELISA and immunohistochemistry. We rely on these types of assays every day for direct measurement of endogenous unmodified proteins. We oftentimes also curse and then work to improve the immunoreagents that we have accessible to us. And of course, there's been a lot of work in understanding some of the limits to the performance of those immunoreagents, and some excellent work moving us forward to more reliability with use of those immunoreagents. Here is, if I don't have an immunoreagent that is selective to my proteoform of interest, I cannot specifically measure that proteoform. And we'll talk about this area, as this is where my group is really burrowing in to try to move the power of immunoreagents and immunoassays forward by using some approaches that take advantage of microfluidic systems. I'll also make a note here that background can be an issue, background signal. When we think about all that we do to the cells and the epitopes on the targets of interest, when we need to do sample prep like chemical fixation, it also becomes an issue with target multiplexing as well. So that's a little bit of context for where the gap is. The work I'll talk about today really looks at how can we enhance this area of immunoassays, single cell or subcellular resolution for the detection of not just proteins, but more specific, the stoichiometry of proteoforms that are being expressed. We look at things and we do optimization and try to bring forward new approaches to enhance the target specificity. That's the key for today's talk. Also, of course, detection sensitivity and moving to subcellular resolution is one example of that. Sample throughput. And then I will mention a couple of times, as designers, measurement scientists, and engineers, we're very attuned to design for translation. And that means understanding how tools are used, be they in a clinical setting, in a life sciences research setting, or in the field, and trying to develop technologies that can be adopted easily into those settings. So human-centered design, appropriate for the location of use. So just to dive in now to the approaches that we're using that I'll walk you through with a couple of examples. The tools, again, use microfluidic design principles. We're designing those tools for single cell and subcellular resolution. We're designing those tools to capture molecular heterogeneity, but with rapid, so efficient and sensitive separations of those molecules. And then one of the aims of this work is to harness and bolster these less than ideal but powerful immunoreagents. I will say the designs I'll show you today are agnostic to the immunoreagents that we use. So the examples I'll give typically use antibodies for probing of targets of interest, but that need not be the case. Other binding reagents are equally amenable to the system design described here. So the approach that I'll talk about brings together a compartmentalization methodology. So we typically use microwell arrays to isolate, manipulate, and then handle individual cells. Other compartmentalization approaches that you may be familiar with include droplets and microchambers. We like the microwells for the assays I'll describe today. We append those microwells with a two-stage assay. So using a separation stage to gain information about a specific physicochemical property of interest. And then, as the foreshadowing in my earlier slides mentioned, we concatenate that separation stage with a probing stage. And the examples I'll show today are an immunoassay. So this combination of separation and probing is known more widely as immunoblotting. If you do life sciences research or even clinical medicine, you may have used one of the most maybe well-known types of immunoblot known as a Western blot that concatenates together protein sizing with its immunoprobing immunoassay step. Of course, there are literally tens to hundreds of different types of separations, not just protein sizing, that we can use to really optimize the single cell immunoblot to the physical chemical property of interest. So that's one of the amazing aspects of separation science, to be able to really tune that to the question at hand. So microfluidics allows us to bring precision control of the chemistries that we're using both in space and time. I would say arguably one of the biggest advantages of using a microfluidic approach is integration. So to be able to tie together nearly seamlessly and almost instantaneously different types of function, including sample preparation, and in what I'll talk about today, this tandem assay, the separation coupled with the immunoprobing step. So hopefully you'll see some examples of that, and I'll try to highlight where integration makes possible some performance that isn't possible on the macro scale. So as a brief outline, the overall theme for the work I'll share is designing these high specificity single cell resolution analysis tools here with an emphasis on cancer research. 3 examples that I'll provide. One is looking at truncated isoforms, in the particular case of ERBB2, HER2, in dissociated solid tumors. The second example is going to be some work with scarce starting cell populations, so protein profiling of single circulating tumor cells. And then the last aspect is going to be looking at developing a proteoform atlas using intact cell layers or tissues. And again, that is the topic of the projection electrophoresis that was mentioned earlier. So this is what a typical device from my research group looks like. This is a microfluidic device, but it actually looks a little bit more like a microscope slide, and I'll talk about why that's important in a minute. But just for orientation, this is an isometric view here, a little bit of a perspective view. You see here the thickness of a microscope slide. So it's a standard microscope slide that's been silanized, the surface treated, so that we can cast a thin layer of a hydrogel. And in everything I'll talk about today, that's a polyacrylamide gel, which is, of course, the gels that are widely used for protein electrophoresis and immunoblotting. So there's a good connection there in terms of being able to do microfabrication with this type of material and to be able to get the pore size in those hydrogels to allow molecular sieving to be effective for the size of the targets of our interest, which are proteins. We've got that thin layer of gel, typically 30 to 100 microns thick. And for context, a human hair diameter is roughly 100 microns in thickness. The other feature that I'll point out are these arrays of microwells and abutting each microwell a separation lane. So you can see an array of about 420 microwells here, a confocal image here of a single neural stem cell sitting inside of one of those microwells that's formed by the hydrogel. So just a little bit of context. Something you won't notice here, we don't have any pneumatics at all on the device. And by that, I mean there are no pumps or valves on this device. It is a passive device for all intents and purposes to the end user. And I'll talk about how we encode function without impinging upon the end user's use of the device very much. So it is a passive device, no pumps or valves that are included. And you're also going to notice there's no real precision design here, except for the microwell arrays. There's no electrodes, densely patterned electrodes. It is a pretty simple structure. And the reason for that, again, is design for translation. We'd like to do everything we can in the materials and the physics, the control of the physics, and take all of that precision, manipulation, and effort out of the hands of the end user. Okay, so what does this assay look like? How do we do this single cell resolution measurements of these proteoform targets of interest? So using a device like I just showed there, we've adapted immunoblotting technologies using a 4-stage workflow. So just for context, on the left, you have a false color confocal micrograph, again, of the microwell, 2 GFP expressing mammalian cells, and we've added some dye to the polyacrylamide so that you can see that. We settle typically single cells into these compartments, into the microwell. We also do active seeding using centrifugation and microtransfer pipettes. I'll talk a little bit about that. But when we have a large population of cells that we want to analyze, we use just gravity to be able to settle those cells in the microwell. That results in something similar to what you would see with a droplet-based system, so typically Poisson statistics. Some of our microwells have no cells in them. Some of our microwells have 2 cells in them, and most of the microwells have 1 cell in them based on the design of the microwell itself, and in particular, the aspect ratio of the microwell. If you're interested in more information, we have in this Nature Protocol article really explicitly defined all of the engineering constraints and design considerations for developing a compartmentalization system using microwells like described here. Next, we apply an electric field across the hydrogel. So this region right here is the hydrogel that I mentioned, the polyacrylamide gel. It's structural. It allows us to form the microwell, but then it's also functional because it allows us to, under the application of that applied electric field, electromigrate solubilized species from cell lysate. So we've lysed the cell in the microwell here into the polyacrylamide. So that gel acts like a molecular sieving matrix, allowing us to resolve targets based on differences in their size, differences in their charge, or differences in their isoelectric points, among other things. And the typical single cell lysate that we're analyzing is about 30 picoliters in volume. So that's the separation stage. To move to the second stage of the assay, so I mentioned immunoblot, separation, tuned to the physicochemical property of interest, but coupled with an immunoassay. And this is important because we now have 2 pieces of information, the separation output and the immunoassay output, which gives us more selectivity than the immunoassay alone. And that's the whole reason for pretending the separation stage to the immunoassay. But we need to toggle. We need to toggle now between that separation and that immunoassay. And here in the separation stage, our targets of interest have electromigrated through the gel, but those molecular species, those proteins in this case, are in solution in the buffer solution that is in the pores of the hydrogel. So this is called a homogeneous system because the proteins are in solution in the hydrogel pores. But to move to an immunoassay, we need to toggle to a heterogeneous system, which means we need to somehow lock down those proteins to a substrate, either a surface or, in our case, the 3D hydrogel. So the way that we've opted to do that is to include a photoactive moiety in the polyacrylamide gel. So here we're using benzophenone methacrylamide. It's part of that cross-linked polymer when we fabricate it. But when we shine UV light onto the gel after the separation stage, we abstract hydrogen from any CH bonds that are present near that benzophenone, and we covalently attach those species to the hydrogel matrix. So here, just use light, a brief exposure, 40 to 60 seconds, to toggle between a separation and an immunoassay, at which we've brought in the probe of interest and probe for our target. So 2 pieces of information that come out of this assay and hence the higher selectivity. What does this look like? So again, one of our open devices. This is an example of one of our first approaches using this technology. So about 6,700 single cell, in this case, Western blots on one of these devices. Again, that open fluidic design, so it pretty much operates like a microscope slide to the end user. Here we're looking at neural stem cells. That's not really the important part. What I did want to show you is what does a readout from a single cell immunoblot look like. So this is a false color micrograph, fluorescence micrograph of those microwells you can see and the abutting separation lane. So you've got your bright field here. You can see that distribution of cell numbers, the microwell occupancy, and then abutting each of those cells is the endpoint readout. Here we've just probed for 2 targets of interest, beta tubulin and GFP, to show the example of what this sort of single cell Western, in this case, looks like. The key feature to note, we have about 500 microns of separation distance, so orders of magnitude smaller than what you would expect on a conventional Western. Obviously, this is tuned for a single cell analysis and not a bulk sample. In addition to that, the separation itself, the electrophoresis stage, is typically 15 to 30 seconds in duration, so exceptionally short, followed by the immunoprobing. And using fluorescence data like this, we can back out area under the curve and the electromigration characteristics of each species to estimate molecular mass, for example. And so we can treat these, as analytical chemists do, as quantitative and semi-quantitative output regarding that endogenous protein expression in each of the cells. We've done quite a bit of work to really get a handle on the dynamic detection range and the lower and upper limits of detection. I won't go into it today. I'll just look over here on the left, and suffice to say that we see detection limits that are on par with fluorescence cytometry, flow cytometry. So that's an excellent sanity check because our assay is not that different from flow cytometry, except there's a lysis and separation stage before the fluorescence readout. So we did want to check to see we're on par with that. We're estimating about a lower limit of detection, about a femtogram of each of the targets of interest. And for us right now with the tools I'll show today, this puts us in about the top half of the mammalian proteome. So the top 50% most expressed proteins. We have other approaches that really try to push us into those more lower expression species, the bottom half of that proteome. I did want to make a comment on two things related to translation, since I emphasized it a little bit at the beginning of the talk. We've had a couple of spinouts from my research group, and this one is relevant to the technologies I'm describing today that I've just described. So those aspects have been commercialized by Zephyrus Biosciences. It was founded in 2013. I was a co-founder with an amazing alumna from my research group, Kelly Gardner. That startup was acquired in 2016. I no longer am associated with that startup at all, but the really, I guess I would say very useful, fulfilling, and interesting aspect is seeing something that was developed in your lab now sold worldwide, to both understand how people use it and don't use it, and also to understand kind of what aspects of the design make it something that's amenable to large scale use. That's one shout out. The second I did want to say is we are committed to translation broadly. And so of course that means commercialization when that makes sense. But it also means knowledge translation, obviously through publications. But we spent about 6 years in residence in the summers, a short period each summer, teaching graduate students, postdocs, faculty, and clinicians how to make, use, and analyze these types of devices. So I've been fortunate to be part of the Cold Spring Harbor Lab summer courses to do this. This is an alumna from my group who is featured in Nature Methods as she's teaching one of our cohorts how to fabricate, use, and analyze the data. So with projects like this and programs like this NIH program, we see it as an imperative to us, but also very useful to understanding researchers' questions to translate our tools out into the world. And this is one way we do that through the Cold Spring Harbor Lab and similar courses as well. Okay, I will dive into the first example, which is really around these truncated isoforms in HER2, in this case in solid, dissociated solid tumors. So at the beginning, I mentioned a little bit about HER2. Again, a large 185 transmembrane protein with this important extracellular domain. I did want to take a moment here to highlight the fact that we expect not just a single truncated isoform of HER2, but the potential, the possibility of a multiple different versions of the truncated isoform, and also different locations in the cell. So the plasma membrane, the cytosol, and also in the nucleus as well. So that's important. I've been mentioning the stoichiometry of proteoforms. This is an example of a question where we would want to understand the stoichiometry of these forms. Again, questions about drug resistance, development of drug resistance, as well as outcomes, patient outcomes. In HER2 positive breast cancer, the expression of these truncated isoforms has been associated with shortened times of progression-free survival. So just wanted to be more specific on that today. This is two of the forms, proteoforms of HER2. We started developing our tools to measure, to distinguish. So on the right here, this is again, false color fluorescence micrograph of a BT474, so a model system, single cell that we know overexpresses this 185 kilodalton protein. So this is a single cell immunoblot, and you're looking at the fluorescent signal that's coming from probing with an antibody, primary and secondary, the secondary labeled fluorophore here. We began the validation process by, of course, challenging the assay with cells that are engineered with known protein expression profiles. And in this case, we were able to analyze cells that are known to have the large proteoform, transmembrane proteoform, and then cells that are engineered to have that, as well as this smaller truncated form. So this is about 110 kilodaltons versus 185. And on the right, what you can see is this ability to distinguish between the full length and the truncated form of HER2 using a single HER2 antibody. So that's really the key here. The reason why we do immunoblots, whether they are single cell or not, is because I can use separation in space, which is dictated by my choice of separation mechanism and the physical chemical property I'm interested in measuring here, size differences, but use a single well-validated antibody probe to be able to detect and then that separation to distinguish between the two. Again, I mentioned today I'm going to keep this a little bit focused to kind of show the suites of technologies where we think microfluidic design can really help move forward this understanding of cellular to molecular level heterogeneity. But we're also pursuing questions around other truncated isoforms, including estrogen receptor. And I just put an example here of a couple of different where we've analyzed and been able to detect the different stoichiometry of, in this case, estrogen receptor isoforms that are being expressed. The next question for us is really around target multiplexing. So being able to look at a wide range of targets, in some cases that have very large molecular mass and small molecular mass. So the hard part here isn't distinguishing large from small, but when you have large from small, distinguishing too small or too large from each other. So these pairwise separations are somewhat difficult to perform. So I'm just showing here two cells, again, around this question of HER2, where we've analyzed for that target, so being able to detect truncated isoforms in certain cells, but then be able to add in a whole sequence of additional protein targets allows us to link that HER2 stoichiometry to the signaling state of the proteins that are present as well. So this is some 10-plex multiplexing. The key here is not so much how high can we go in terms of the target multiplexing, but the fact that we can do multiplexing that includes proteoforms in that. So that's really the key. We've also started to look at different readout modes beyond fluorescence, and I'll just put this up briefly. Just accepted article, so if you're interested in more information, please let me know. But looking at things like CyTOF, or in this case, multiplex ion beam imaging to be able to read out these types of separations, and you can see a separation here in fluorescence and with the MIBI readout to give us perhaps more power in terms of doing multiplexed target detection in the long run. And then in this case, we've opted to use small gradient gel. So this is one of our devices. Again, a bright field image of the device. These are fingers holding that open fluidic device here, the array of microwells, and then each separation lane, we've added a dye so that you can see the change in gel density as we separate the species from top to bottom. And that really allows us to separate small species from each other, smaller species from each other, and larger species from each other, all in the same separation lane, which in this case is about 1 millimeter. So again, it's pretty short separation distances, which makes this challenging. When we have analyzed patient-derived solid tumors, we've asked questions about at the single cell level, what does the expression of these truncated isoforms look like? So here we've compared two solid tumors dissociated for the full-length protein expression and then the expression levels of the truncated isoforms. And in this case, we see appreciable correlation between the truncated isoforms and the HER2 expression in each cell, which is consistent within the tumor, which is consistent with what's been observed previously. And this lends some additional credibility to the hypothesis that at least a fraction of these truncated isoforms may originate from this protease shedding and not just alternative splicing as well. So that's an open question right now too. Here, I'll just highlight the fact that among, in this case, just two tumors that we were analyzing the truncated isoforms versus the full-length isoforms, we do see statistically different expression of that ratio, the truncated to the full length. So there is a lot of heterogeneity here at the tumor to tumor level, as well as the cell to cell level, and obviously the molecular level as well. So that is important to note. I'm going to skip just a couple of things to mention that we are working on tools for looking at subcellular localization. So this was something that was important to this question of HER2, this not just the stoichiometry, but where is the protein and what protein form at the time of function. And so for us, that means things like concatenating our system to FACS so that we can do enrichment for specific surface receptor expression. So this is immunoblotting that's done, performed on a set of cells that have been eluted by FACS based on enrichment there. In addition to that, really using chemistry, not image segmentation approaches, which are powerful, but somewhat subjective. Here using chemistry to be able to differentially fractionate an individual cell. So using differential detergent fractionation to lyse as a cell sits in the microwell of the outer membrane of the cell, analyze the contents of the cytoplasm with immunoblotting, keeping the nucleus intact, and then going through lysing that nucleus and then performing immunoblotting on the nuclear proteins as well. So using space here in chemistry to fractionate the nuclear versus the cytoplasmic fraction, which we've started to apply to single mitochondrion as well, which is just put here as a mention, the power of the precision of using these types of microfluidic systems, even when the system itself is handled by an end user without the need for precision handling as well. So I'm just looking at the time. I think what I'll mention here is some work that we're doing to really move into these multi-mode separations. So this bottom sequence here is just the single cell analysis approach for immunoblotting that I talked about in the previous portion of this talk, with, in this case, extraction of nuclei-laden microwells, so keeping that nucleus or group of nuclei intact, popping that off the device, we call that a gel palette, being able to suspend that material and then perform in tandem with the proteomic analysis, the targeted proteomic analysis, measurement of the nucleic acids. In this case, some recent publications looking at RT-qPCR, but also moving to sequencing as well. And of course, that's indexed back to the starting cell. So being able to correlate those together, those different modes of information together is pretty important. Okay, so that's that. I'm going to skip over this. The moderator pops back on. I know it will be just about time to start questions. The last thing I wanted to talk about is really looking at this proteoform atlasing of intact cell layers, and an approach that we've developed that we're calling projection electrophoresis. So the concept here is basically starting with a cell layer or a tissue slice on a 2D plane. And instead of performing the cellular level separations in that XY plane, using the Z dimension as well. So performing electrophoresis in Z, and importantly, being able to use that Z dimension as an analytical dimension, not a preparatory dimension. So by using Z for analytical electrophoresis, that allows us to get to the point where in Z-axis, we can gain information on the proteoforms being expressed and then map those back to that starting XY plane. So we start to get a map or an atlas of the proteoforms. And that's really the add of being able to do that analytical separation in the Z dimension. There's some really nice work that's looked at a Z dimension for fractionation, to be able to take crude fractions, either interacting with layers of immobilized antibodies, or other approaches, but the key here is to actually use the separation power of that Z-axis to get analytical information, analytical grade information about the starting tissue slice or cell sample. So this is what the workflow looks like. You've seen every step of this previously, but now we're just performing each of the steps in the Z-axis in a voxel. And this is a nice image that Samantha Grist, Dr. Grist in my lab took of what the device looks like. So again, a really simple open microfluidic device that allows us, this is some confocal imaging here, to get out information in the Z dimension about what the protein expression in that starting cell layer looks like. Some more analyzed data here looking at both a cytosolic and a nuclear protein. So these two species from an individual cell, this is a fluorescence intensity map in the Z dimension. Here we've just revolved that So we get information about a little bit easier to process in terms of visually looking at the expression levels. At each of these two slices, the planar images are shown here, and then we're able to, of course, back out information on the electromigration, which ties to the electrophoretic mobility, and that, of course, reads out the physicochemical property of interest, be it size or other characteristics. Scale up was an important motivation for us, so being able to keep the context of that cell layer or system, but also being able to analyze, again, thousands of cells at a time. And these are just kind of a montage, if you will, of the revolved fluorescence images for a group of cells that were analyzed in a projection electrophoresis system with, of course, that analytical grade information, that separation coming out from information on the Z dimension there. And this is one just example of we either use confocal or light sheet microscopy to be able to image kind of what these plumes of protein look like, these peaks if they were in a slab gel, but now plumes because they're in a 3D voxel, and relate that back up to the top layer to get a map of expression there. So this is one example for some two well-characterized targets to validate the assay, and in this case, in 2D tissue mimics. Okay, so with that, I think I am just about at time. We wanted to leave some opportunity for questions. I hope I've given you some context for how measurement science and engineering design, especially in this area of microfluidics, can potentially open new doors to measurements that are relevant to the biosciences and biomedicine. Specifically here, we've been looking at high selectivity assays, really optimized towards questions about proteoform, stoichiometry, and expression. Relevant example given was the HER2 and truncated isoforms, again, at the single cell, and I also had the chance to show some subcellular resolution with the analysis of those individual nuclei and mitochondria as well. So with that, I want to just, again, thank my research group, very much thank this program for their support and forward-looking vision in terms of bringing together measurement scientists, analytical chemists, and engineers with life sciences researchers and physician scientists in our case as well. Thank you so much.
Thank you, Professor Herr. It was wonderful and lovely to see the merging of pathology with high resolution molecular tests, especially in the protein domain. So a bunch of questions have already come in, but I wanted to take advantage of my position here and ask a couple upfront from my perspective. Actually, I have three, which is cheating. But one is there a way to adapt other versions of spatial profiling by spatially encoding the cells and then not trying to deal with them with their spatial identity intact, but basically just have a barcode or something on them, and then you can use the most efficient way of looking at them. So that's question one.
Sure, yeah, I very much appreciate that question. I know barcoding is all the rage in terms of being able to map back information, and certainly there is huge roles for that to play. In our case, we are a little bit interested in being able to tie information, for example, from staining that is commonly used on the intact cell layers to the proteoform expression profiles, which we think can be done more directly by keeping the tissues intact. Something I didn't mention here today, but is a big area of interest for us, is mapping back to the matrix and what kind of that local cellular kind of proteinaceous environment looks like. So that's something that may not be so easily captured with barcoding as well. But your point is well taken, that there are some great opportunities for using things like barcoding and then allowing you to dissociate, release, and analyze maybe in an efficient way. But in this case, we think that keeping the information intact allows us to access some of those other molecules and features of interest that might be lost. Great point.
And then I'll take one more question, then I'll hand over. I'll then pull up the other ones. And that is, this is an imaging, basically imaging based, and there are a lot of techniques for AI-based super resolution and denoising and all of that stuff. Have you done anything more than with your images themselves?
Yeah, absolutely. Yeah, so we've been using machine learning. I have just an amazing group of engineers as well as chemists and clinical collaborators as well. But we have been using machine learning to help us with image segmentation to make the analyses more efficient and also less subjective in some cases. I didn't have time to talk about that today, but absolutely, we see those approaches as exceptionally powerful and definitely quite relevant to the work that I showed here. So yes, the short answer is yes.
Oh, excellent. So I have a bunch of questions, 8. Let's see how far we can get with some of them. Gary Davis has asked 2, so I'll, and
Sounds good.
Looks like he's given, someone's giving him two thumbs up. So,
oh, excellent.
So he says the next frontier are kinetic relationships binding, see what happened to it, binding reaction rates, et cetera, at the proteoform level. Basically, you've obviously opened yourself up to all of protein chemistry. And then
what's the next frontier? Is that right? Did I hear you?
Yeah, pretty much that.
Yeah. Thank you so much. Yeah, Gary, I think, I hope I'm using the right name here. That is just a phenomenal question. And the next frontier, I would argue there's a lot to do here, both fortunately from the standpoint of being able to unlock new information, but unfortunately as well. And this is just a little bit of my own personal perspective, which if there are trainees on the call, I would love to emphasize that this area of proteoforms, but also more broadly proteomics, it has a lot of great, just amazing researchers around the world working on the measurement sciences aspects. But I do have to say, I think there's a lot of room for additional innovation, rigor, and creativity. And so I would encourage you to please take a look at this area if you're thinking about your vision for kind of the early stages of your career. But yes, Gary, some big areas that we're pushing for, as I mentioned, this multimodal, so being able to tie together different types of molecular information in a correlated way with single cell or subcellular resolution. But definitely an area that we've been working on for the past couple of years has to do with complex formation. And I think you mentioned that in your question as well, being able to really determine when we have homodimers or heterodimers that are forming, specifically in the case of HER2, can we tease apart the stoichiometry, not just of the proteoforms, but the composition of those heterodimers or homodimers, for example. Do we expect full length and full length HER2 to be partnered together in a homodimer, or could a homodimer also, and I guess this would be a heterodimer, consist of something like the full length and the truncated or different forms of the truncated? So I think there's a lot of rich information that is there. Of course, other researchers started to look at this as well, but that's just one example that really, I think, aligns with your question very well. We're also, of course, interested in activity. So zymography and different types of approaches in that theme are of great interest to us as well, and are compatible with the tools that I've described today. So Gary, thank you so much for asking that question. I appreciate it a lot.
Hey, here's one from David Chen with 6 upvotes. And the question is, can it be combined with single cell RNA-seq and proteoform resolution?
Yes. Yeah, excellent question. So this is along the theme of the multi-mode assays I talked about very, very briefly, and I apologize for just giving the tip of the iceberg in terms of what we're doing, but I think more importantly, what can be done in the field, not just by us, but other labs as well. What we've demonstrated and published so far is concatenation of the proteoform measurements at single cell resolution, as I mentioned, with the RT-qPCR and other PCR-based approaches. We definitely think there is room for tying this together with sequencing as well. Now, the approach that we have been using really is this differential fractionation of the cell into the cytoplasmic component and the nuclear component. So the approach that we're taking right now, some of which is not published yet, is pulling out the nucleus and being able to analyze the nucleic acids that are in the nucleus. So I think an area that really, really would demand some creativity, and it's probably a little beyond where we're at, but certainly I'm interested in going there, is how do you also capture what's happening in the cytoplasm of the cell, not just the nucleus as well. So maybe, I hope that answer is clear, but yes, absolutely. We think that there is the possibility and are working towards that for sure.
Here's another question now on the 3D projection electrophoresis. Tissues are very heterogeneous and tightly packed. And so how do you resolve sort of multiple cell types within a very small region?
Yeah, that is a phenomenal question. I think I'm probably talking to a couple of people in this audience that are much more expert in this than I am. So if you have ideas or if you think there's something that we're overlooking, which I know is a lot, please reach out to me. Again, my email address is at the bottom. I'd love to continue the conversation. We have a lot to learn. But yeah, in terms of that specific question, I would say being able to tie together some of the upfront imaging, I'll call it conventional pathology-based imaging with some of the approaches I talked about here to get that proteoform information is going to be really important to look at. I do really appreciate the aspect about the tightly packed cells. We have noticed a tremendous amount of variability won't be surprised to most of you on this talk for sure, in the audience for sure, a tremendous amount of variability in terms of how we can prep those cells. And in our case, it's primarily around lysis and antigen retrieval if the cell system has been fixed, for example, FFPE or other approaches to fixation. So one of my colleagues would always say, probably the least sexy but most important area of single cell analysis is sample prep. And I have to stand behind him and say, absolutely. And the question that you asked, in my first response to that would be the need to be able to tailor the sample prep for the specific types and regions of tissues of interest. For us, we do have some approaches that we think are promising. I did not talk about them today, but using almost like a hydrogel sticker to be able to pattern on a surface by making contact between that surface and this reagent imbued hydrogel to be able to pattern the types of sample prep. So not just subjecting the entire cell layer to a single type of sample prep, but using spatial patterning of the lysis reagents and applying that to the cell layer allows us to get some more optimization. But excellent point, a lot more work to go. And I hope that answer was specific enough for today's discussion.
Can you distinguish multiple proteoforms or spliced isoforms if they're co-expressed in a single cell?
Yes, I see. Yes, absolutely. So what I showed today is really in the case of HER2 and the ER that I showed, really are proteoforms. Yes, they're co-expressed in the same cell. Definitely. We have, especially in the case of the truncated ERBB2 forms, some of the data that I showed, and I had to go a little bit quick, but some of that is published, did look at the co-expression of the truncated isoforms with the full-length HER2. So yes, you can do that. I think maybe part of your question has to do with more nuance here, and that is the breadth of the stoichiometry. So, for example, can we resolve a 95 versus 110 kilodalton proteoform, kind of with that whole family of proteoforms that I showed in that one schematic on HER2? And the answer there is yes, in certain cases we can. We oftentimes, when we have close molecular mass, especially for large targets, we use another approach that's known as isoelectric focusing instead of the size-based electrophoresis, because we can more readily distinguish those different close in size proteoforms from each other by using differences in their isoelectric point. So again, it kind of speaks to the fact that we can tune the separation stage to answer the physical chemical question of interest with our proteoform targets. I hope that answers your question.
So here's a question that just came in, which I think it's a very apt one. How to apply this technique in routine practice? How would you compare this to traditional immunohistochemistry studies or immunofluorescence used clinically on breast cancer slides, for example? Is this going to replace immunofluorescence under a microscope on whole tissue?
Right. So my answer to that is we've developed these technologies to fill an existing gap. And so specifically to your point, with proteoforms, we are developing these technologies to complement and augment existing tools. So with what I described today, I think that would be probably not realistic to say they're designed to overtake existing tools in clinical practice, but they are designed to be able to unlock information that you can't get out of tools in clinical practice. So certainly, I think there's also potentially the opportunity here as we're thinking about cytopathology and different assays in distributed low resource settings, be they low resource globally or within our own nation, thinking about are there ways to get out more quantitative, reproducible information by using this ability to make precision measurements, but by designing devices that don't require the end user to apply precision actuation to the device. So basically handle it like a microscope slide, but the microscope slide is instrumented with microfluidics that can help with more precision. So the realistic answer to your question is these tools are designed to fill gaps and not to necessarily fully supplant entire classes of assays like immunohistochemistry.
Fair enough. I think we have reached 10 o'clock, and so I would like to thank all of the unanswered question posers and much more so our speaker. This was really a wonderful experience, and I thank you all. On behalf of everyone else, it's not me, Tony, and I think Tony can wrap this up, right, Tony?
Yes, thank you, Dr. Herr and Dr. Levinson, very much for an invigorating talk, really exciting advances. Really appreciate your giving us some insights into how things have moved through, and thank you, Dr. Levinson, for your exquisite moderating skills. And let me just put out for the rest of you on the call, please note that this kind of ends the first season of the Cancer Moonshot Seminar Series, but we're already actively planning the second season. And I just draw everyone's attention to the QR code on the screen. We are accepting nominations for junior investigators to participate in some of these Moonshot project highlights. And so those of you associated with Moonshot funded projects out there that have a junior investigator in mind, please do consider nominating them to contribute to this series. And thank you all so much again. And we'll be back in your calendar soon with new and exciting talks to look forward to in the future. So thank you very much, everyone.